Hello everyone. Welcome to our exploration of reproduction in biology. Today we're going to discover how unicellular organisms and invertebrates create new life and continue their species. Reproduction is the fundamental biological process that allows organisms to create new individuals and ensure the survival of their species. Without reproduction, life would simply end. There are two main types of reproduction. Asexual reproduction involves only one parent and produces offspring that are genetically identical. Sexual reproduction involves two parents and creates genetic variation in the offspring. We'll examine several fascinating organisms. Among unicellular organisms, we'll study Amoeba, Paramecium, and Spirogyra. For invertebrates, we'll explore earthworms, cockroaches, and houseflies, each with unique reproductive strategies. You'll see detailed processes like binary fission in Amoeba, where one cell divides into two identical cells, and conjugation in Paramecium, where two cells exchange genetic material. These amazing processes show the diversity of life's reproductive strategies. Get ready to explore the fascinating world of reproduction. We'll start with the simplest organisms and work our way up, discovering how life finds amazing ways to continue and thrive. Amoebas are fascinating single-celled organisms that reproduce through asexual reproduction. This means they can create identical copies of themselves without needing a partner. Here we see the structure of an amoeba. It's a single-celled organism with a flexible, ever-changing shape. The nucleus controls all cellular activities, while pseudopodia help it move and capture food. The primary method of reproduction in amoebas is called binary fission. Think of it as the amoeba making a perfect copy of itself by splitting into two identical halves. The binary fission process follows a specific sequence. First, the nucleus elongates and begins to divide. Then the entire cell starts to constrict in the middle, like pinching a balloon. Step 1. The nucleus, which contains the amoeba's genetic material, divides into two identical nuclei. This ensures each new amoeba will have the same genetic information. Step 2. The cell membrane begins to pinch inward from opposite sides, gradually separating the cytoplasm and organelles between the two forming cells. Step 3. The division completes, resulting in two identical daughter cells. Each new amoeba is genetically identical to the parent and can immediately begin living independently. When environmental conditions become unfavorable, such as during drought or extreme temperatures, amoebas have another reproduction strategy called multiple fission. During multiple fission, the parent amoeba forms a protective cyst around itself. Inside this cyst, the nucleus divides multiple times, creating many small nuclei. The cytoplasm then divides around each nucleus, forming many small amoebas within the cyst. When conditions improve, the cyst breaks open and releases all the new amoebas. Here's a complete overview of amoeba reproduction. Remember these key points. Amoebas reproduce asexually, creating identical copies through binary fission under normal conditions and multiple fission when stressed. This asexual reproduction strategy allows amoebas to rapidly increase their population when conditions are favorable, making them highly successful single-celled organisms in various environments. The Paramecium is a fascinating slipper-shaped unicellular organism that demonstrates remarkable reproductive flexibility. Unlike simpler organisms, Paramecia can reproduce in two distinct ways, giving them unique advantages for survival. First, let's examine asexual reproduction through binary fission. This process begins when the Paramecium reaches maturity. The nucleus divides first, followed by the elongation and constriction of the cell body, ultimately producing two genetically identical daughter cells. Sexual reproduction in Paramecia occurs through a remarkable process called conjugation. Two Paramecia come together and form a temporary bridge, allowing them to exchange genetic material. This process doesn't produce offspring immediately, but it creates genetic diversity that benefits future generations. Having both reproductive methods gives Paramecia significant advantages. Binary fission allows for rapid population growth when conditions are favorable, while conjugation provides genetic diversity that helps populations adapt to changing environments and survive challenges. The key takeaway is that Paramecia showcase remarkable reproductive flexibility. They use binary fission when rapid population growth is needed, and conjugation when genetic diversity becomes important for long-term survival. This dual strategy makes them highly successful microorganisms. Spirodra is a fascinating type of green algae that lives in freshwater environments. This filamentous organism has two distinct methods of reproduction that we'll explore today. First, let's understand what Spirodra looks like. It forms long, thread-like filaments made up of cylindrical cells connected end to end. Notice the spiral-shaped chloroplasts inside each cell. This is where Spirodra gets its name, meaning spiral thread. Each cell contains a nucleus, cytoplasm, and other organelles needed for life. Spirodra reproduces in two main ways: asexual reproduction through fragmentation, and sexual reproduction through conjugation. Fragmentation is the simpler, asexual method. When environmental conditions are favorable, the Spirodra filament simply breaks apart into smaller pieces. Each fragment contains several cells and has the ability to grow into a completely new filament. This process is very efficient and produces offspring that are genetically identical to the parent. Now let's explore conjugation, which is Spirodra's method of sexual reproduction. This process is more complex but creates genetic diversity. Conjugation occurs when two Spirodra filaments lie side by side. Special tubes called conjugation tubes form between corresponding cells of the two filaments. Through these conjugation tubes, the contents of one cell, acting as the male gamete, transfer into the neighboring cell, which acts as the female gamete. When the gametes fuse, they form a thick-walled structure called a zygospore. This zygospore can survive harsh environmental conditions and later germinate to produce a new Spirodra filament. Let's compare these two reproduction methods. Fragmentation is quick and simple, producing many identical offspring when conditions are good. Conjugation, while slower, creates genetic variation and produces hardy zygospores that can survive when environmental conditions become difficult. Both methods ensure Spirodra's survival: fragmentation for rapid population growth in good times, and conjugation for genetic diversity and survival during challenging periods. Now we move from unicellular organisms to invertebrates. Earthworms represent a fascinating example of sexual reproduction in the animal kingdom. Unlike the unicellular organisms we studied earlier, earthworms reproduce sexually. What makes them unique is that they are hermaphrodites, meaning each individual earthworm has both male and female reproductive organs. Here we can see the anatomy of an earthworm. Notice the clitellum, which is a specialized thickened segment that plays a crucial role in reproduction. This structure secretes the cocoon, where eggs will be fertilized and develop. The copulation process in earthworms is quite remarkable. Two earthworms come together and align themselves in opposite directions, creating a head-to-tail arrangement. During this intimate contact, they exchange sperm with each other. This diagram shows the three main stages of earthworm reproduction after copulation. First, the clitellum forms a girdle around the worm's body. Then, the worm moves backward, and finally, a protective cocoon is created containing the fertilized eggs. The cocoon serves as a protective environment where fertilization actually takes place. The eggs are fertilized by the sperm that was exchanged during copulation and stored in the worm's body. Young earthworms develop safely inside this cocoon and hatch after about 2 to 3 weeks. Sexual reproduction in earthworms provides several important advantages. It creates genetic diversity in offspring, which helps populations adapt better to changing environments and increases their overall survival chances compared to asexual reproduction. To summarize earthworm reproduction, they are hermaphrodites with both male and female organs. Two worms exchange sperm during copulation. The clitellum creates a protective cocoon, and fertilization happens safely inside this cocoon. This sexual reproduction strategy ensures genetic diversity and species survival. Cockroaches are fascinating insects that reproduce through sexual reproduction with internal fertilization. The male cockroach introduces sperm directly into the female's reproductive system. After mating, the female cockroach lays her fertilized eggs in a protective case called an ootheca. This horny egg case shields the developing embryos from environmental dangers. Now let's examine houseflies, which also reproduce sexually through internal fertilization. Female houseflies seek out moist environments to lay their eggs. The eggs hatch into larvae, called maggots. These maggots are the juvenile form of houseflies and feed on organic matter before transforming into adult flies. Snails have a unique reproductive strategy. Most snails are hermaphrodites, meaning each individual has both male and female reproductive organs. During mating, two snails exchange sperm with each other. The eggs are then fertilized internally, and the female lays the fertilized eggs in a cool, dry location for protection. These three organisms demonstrate different strategies for sexual reproduction. Cockroaches protect their eggs in cases, houseflies lay eggs in moist environments, and snails use their hermaphroditic nature for efficient mating. Metamorphosis is one of nature's most fascinating transformations. Many invertebrates undergo dramatic changes as they develop from young to adult forms. There are two main types of metamorphosis that we need to understand. Complete metamorphosis involves 4 distinct stages: egg, larva, pupa, and adult. The larva looks completely different from the adult and undergoes a dramatic transformation inside the pupa stage. Houseflies are another excellent example of complete metamorphosis. The maggot larva looks nothing like the adult fly and must go through the pupa stage to transform completely. Incomplete metamorphosis has only 3 stages: egg, nymph, and adult. The key difference is that the nymph already resembles a miniature version of the adult, just without fully developed wings or reproductive organs. Now let's compare these two types of metamorphosis side by side. Complete metamorphosis involves a dramatic transformation with 4 stages, while incomplete metamorphosis shows gradual development through 3 stages. The key differences are clear. Complete metamorphosis has 4 stages with a dramatic transformation, while incomplete metamorphosis has 3 stages with gradual development. Understanding these patterns helps us classify and study different invertebrate species. Now that we've explored reproduction across different organisms, let's review the key concepts we've learned about how life perpetuates itself through diverse reproductive strategies. First, let's recap asexual reproduction in unicellular organisms. These single-celled life forms create identical copies of themselves through various methods. Amoeba reproduces through binary fission, where the nucleus divides first, followed by the cytoplasm, creating 2 identical daughter cells from one parent cell. Hydra uses fragmentation, where the filament breaks into pieces, and each fragment grows into a new complete organism. Paramecium can reproduce both asexually through binary fission and sexually through conjugation, where 2 cells exchange genetic material. Now let's review sexual reproduction in invertebrates, where organisms combine genetic material from 2 parents to create offspring. Earthworms are hermaphrodites with both male and female organs. They exchange sperm during mating and lay fertilized eggs in protective cocoons. Snails are also hermaphrodites that exchange sperm and lay eggs. They have complex internal anatomy adapted for reproduction. A crucial concept we learned is metamorphosis, the transformation from juvenile to adult form. There are 2 main types. Incomplete metamorphosis has 3 stages: egg, nymph, and adult. The nymph looks like a small adult. Cockroaches follow this pattern. Complete metamorphosis has 4 stages: egg, larva, pupa, and adult. The larva looks completely different from the adult. Houseflies follow this pattern. These diverse reproductive strategies demonstrate the incredible adaptability of life on Earth. Each method has evolved to suit specific environmental conditions and survival needs. Remember these important points. Asexual reproduction creates identical offspring quickly, while sexual reproduction creates genetic diversity. Metamorphosis allows organisms to exploit different resources at different life stages. Now test your understanding with these evaluation questions. Can you define asexual and sexual reproduction? Can you describe reproduction in Spirogyra and Paramecium? Can you explain the difference between complete and incomplete metamorphosis? Understanding these reproductive strategies helps us appreciate the amazing diversity of life and how organisms have adapted to survive and thrive in countless environments across our planet.